Welcome to the Institute of Advanced Studies Li Gongjian Distinguished Professor Public Lecture. I am Song Kiet from the NTU School of Chemistry, Chemical Engineering and Biotechnology. It is my esteemed honor to be the MC for today's public lecture. The lecture is organized by the Institute of Advanced Studies at Nanyang Technological University with support from the Li Foundation. Given the latest COVID guidelines, the event has been made possible in a hybrid mode. We are happy to welcome our audience here at the Tan Chin Tuan Lecture Theatre. We also welcome those who are joining us on the live stream. To begin with, may I now invite on stage Professor Chian, Director, Institute of Advanced Studies, to deliver his welcome address. Professor Sam, please.
Professor Joseph Song, Dean, NTU Li Gongjian School of Medicine and Senior Vice President, Health and Life Sciences. Professor Virginijus Siksnys, Kavli Prize Laureate in Nanoscience 2018. Distinguished guests, colleagues, students, ladies and gentlemen, good afternoon. It is my pleasure to welcome you to the Institute of Advanced Studies Li Gongjian Distinguished Professor Public Lecture at NTU. I would like to first convey my gratitude to our distinguished speaker, Professor Virginijus Siksnys, who is the Chief Scientist at the Institute of Biotechnology, Vilnius University. Thank you for being here with us today and delivering the public lecture at NTU. Now, inspired by a 2007 paper describing the adaptive immune system provided by CRISPR-DNA sequences in bacteria, Professor Siksnys began exploring the role of the enzyme Cas9 in CRISPR. Since then, Professor Siksnys has made a major and sustained contribution to the understanding of the structure and function of restriction enzymes. Now, his current research on CRISPR system for antiviral defense has had a major impact on the field, paving the way for the development of novel tools for genome editing applications. For his exceptional achievements in the biomedical sciences, Professor Siksnys has been recognized with several major awards like the Lithuanian National Science Prize, the Warren Alpert Foundation Prize, and the Kavli Prize. Now, the prestigious Kavli Prize is awarded every 2 years, and it honors scientists for their breakthroughs in 3 fields: astrophysics, nanoscience, and neuroscience. Now, these fields respectively represent the largest, the smallest, and the most complex fields, with exciting possibilities for the 21st century and beyond. A fun fact: it is great coincidence that both the Kavli Prize and the Institute of Advanced Studies at NTU were established in 2005. The hallmark of the Institute of Advanced Studies at NTU is science at the highest level. Now, I'm proud that over these years, IAS has enabled our researchers, students, and faculty members to interact with world-class scholars and scientific leaders around the world like Professor Siksnys, through our cross-disciplinary public lecture series and frontier conferences. Now, I'm sure all of you are looking forward towards this insightful talk by Professor Siksnys on the treasure trove of DNA scissors, and are excited to learn more about the state-of-art developments in CRISPR-Cas and targeted genome engineering. Now, I wish you all a fruitful afternoon. Thank you.
Thank you, Professor Sam. May I now invite our guest of honor, Professor Joseph Sung, Senior Vice President and Dean of the Li Gongjian School of Medicine, on stage to give his opening speech. Professor Joseph Sung, please.
Professor Virginijus Siksnys, distinguished guests, colleagues, ladies and gentlemen, a very good afternoon to all of you. First of all, it is my pleasure to introduce our speaker and moderate this session, because we just witnessed the born of a new school formed by the College of Science and the College of Engineering, the CCEB, 2 hours ago. But instead of asking the Dean of Science or Dean of Engineering to chair this, you asked the Dean of Medicine to come to chair your distinguished lecture, and that is quite an honor for me. Well, I'd like to once again welcome our distinguished speakers on behalf of NTU to deliver today's IAS Li Guangqian Distinguished Professor Public Lecture. Virginijus is currently a professor at Vilnius University, as well as the chief scientist and head of Department of Protein Nucleic Acid Interaction at the Vilnius University Institute of Biotechnology. For close to 3 decades, Virginijus has focused on studying the structure-function relationship of enzyme involved in nucleic acid metabolism. His laboratory in Lithuania has performed many seminal studies of protein involved in bacterial antiviral defense, including restriction endonucleases that are widely used in cloning today, and of course, the CRISPR-Cas system that is really revolutionizing the practice of medicine in the form of novel gene therapy. Well, the concept of gene therapy is not new. Ever since the 60s, humankind are trying to use virus to test the hypothesis that if we introduce a foreign DNA piece into the mammalian cell, whether we can provide new function or even healing some diseases. Subsequently, with the arrival of recombinant DNA technology in the 80s, cloned genes become a reality, and we're able to demonstrate that foreign genes could indeed correct genetic defects and overcome diseases phenotype in mammalian cells, like cells that are growing on a petri dish. However, for many years, gene therapy relies really on random viral integration. This is a serious issue because viruses can insert themselves into some important parts of our genome and disrupt, for example, the good and the proper function of human cells. Indeed, there has been several incidents, including the one in 2003, 2 children received gene therapy for severe combined immunodeficiency syndrome, SCID, and unfortunately developed leukemia. So the obvious solution to address this issue of random viral integration is to find a way to perform precise surgery on our genome. The development of gene targeting by homologous recombination is an early breakthrough in genome manipulation. In this method, the researchers provide a repair template that contains the desired edits to incorporate into a target site. Unfortunately, it produces highly precise alteration in cellular DNA, but the desired recombination event occur very infrequently. Another breakthrough comes in the 90s, where a series of studies demonstrate that target DNA cleavage could greatly stimulate homologous recombination event. Consequently, the search began for an ideal pair of molecular scissors that could readily program to specifically cut a desired location in our gene. I'll shorten the story because the focus today is really on CRISPR. In nature, CRISPR-Cas system function to protect bacteria and other organisms against viral infection and foreign genetic elements. Today, researchers have successfully adapted them for genome and transcriptome engineering purposes. A key reason why CRISPR has garnered so much attention is that its mode of recognizing a target site is simply using a short piece of RNA, which is straightforward to use and can be designed. This ease of manipulation opens the floodgates to a host of biomedical and biotechnological applications that previously would have been challenging to implement, including the use of gene therapies. However, despite tremendous potential of CRISPR technology, we need to be mindful that the ethical issues associated with genome editing cannot be ignored. I remember a few years ago, out of a sudden, there was a technologist in Shenzhen, China, trying to incorporate certain gene to repair the genome of a HIV-infected fetus, and that caused a huge ethical controversies. So much of the ethical debate has centered around the human germline because editing changes introducing the germline will be passed down to future generations. We, as a society, need to deliberate carefully the pros and cons of engineering human embryos and put in place a robust regulatory framework for the application of genome editing technologies. We are very honored today to have with us one of the pioneers of CRISPR, Virginijus, to share his discovery of this intriguing antiviral defense system. Among his numerous achievements, Virginijus was one of the first to demonstrate in 2012 programmable DNA cleavage by the new famous CRISPR-Cas9 protein. He has received many honors. You have just heard from Professor Sum. That include 2016 Warren Albert Foundation Prize, 2017 the Novozymes Prize, and 2018 the Kavli Prize in Nanoscience, among many others. He is also an elected member of AMBO. To the young scientists and students on site and online, I'm sure today you will leave this hall with fully inspired ideas from Virginijus. I encourage you to see how fundamental basic science can be found its own way to impact human life. In closing, I would like to thank the Lee Foundation for its support in this lecture and the Institute of Advanced Studies for organizing this event. I would also like to convey my gratitude to Virginijus for joining us in person to deliver this lecture. Ladies and gentlemen, I present to you Professor Virginijus Siksnys.
Frozen? Oh, okay. No, much better. Okay. So thank you again for kind introduction and also for Lee Foundation for supporting this talk. It's my great pleasure to be here. It's my first time in Singapore and I enjoyed it very much. Yeah, so actually I belong to Vilnius University and my lab is located at the Life Science Center of Vilnius University. And of course, many of you probably are not familiar where Vilnius University is and where Vilnius is. So I decided just to show a few slides. And actually Vilnius is a capital of Lithuania, and Lithuania is one of the Baltic countries. It's located on the shores of the Baltic Sea, and Sweden is just across the sea, and Poland is our southern neighbor. So Vilnius is actually a UNESCO heritage site and probably most famous for the number of churches per square meter. But it doesn't mean that actually we go to church to pray every morning. Just across the river, there is a modern city. And in Vilnius, history and modern times usually live peacefully together. So Vilnius University is one of the oldest universities in the region. So it was established in 1579. And as I said, very recently we moved into a brand new life science center that was launched in 2016. And this life science center is located at the new university campus. So a few years ago, Vilnius appeared on a different map. I borrowed this map from the review by Eric Lander that was published in Cell in 2016. And Eric called this map the CRISPR map of key discoveries. And I was really happy to find Vilnius on this map. So probably it's not surprising that today I will be mainly focusing on CRISPR. So if you look at that timeline of CRISPR, so the CRISPR or CRISPR repeats were discovered in 1987, and actually this discovery culminated in Nobel Prize that was awarded in 2020. But I think that one of the most important dates on this timeline is 2012, when we and others have shown that, in fact, the Cas9 protein that is present in some CRISPR-Cas systems actually is a key player in CRISPR, in the interference provided by CRISPR against invading foreign nucleic acids. And then actually we and others demonstrated that Cas9 could be programmed to cleave DNA at specific locations. And this started a genome editing revolution. So therefore, actually, from my own perspective, the 2012 is time zero. So then actually the time BC, it means before Cas9, and the time AC is after Cas9. So this is my timeline. And yeah, today, in the first part of my talk, I will briefly introduce you how the CRISPR systems were discovered and try to highlight our contribution in that. So you know that immune system in our bodies, so it protects us against invading pathogenic microorganisms and viruses. And immune system is comprised of two arms that are called innate and adaptive immunity. And these two different types of immunity differ by molecules and cells that comprise these immune systems, and also by response time. So the innate immunity can recognize invaders immediately and react to invading viruses immediately. In the case of adaptive immunity, it has needs some time to develop, but then actually it can memorize the invader and actually destroy the invader during the next round of the infection. So bacteria, including pathogenic bacteria, they have their own enemies, and viruses or bacterial viruses or phages, so they are major parasites of bacteria, and phages infect bacteria in order to replicate, and often they kill bacteria when replication is completed. So in this respect, phages represent a lethal threat to bacteria, but bacteria still thrive and multiply. And this is because bacteria during evolution was able to build different defense barriers that interfere nearly with every cycle of virus life. And then, since getting into the bacteria, it's key during phage life cycle, so phages are constantly evolving, trying to break the existing defense barriers, and this forces bacteria actually to develop new defense barriers, and actually this kind of arms race between bacteria and viruses is going on and on. So, and the genes that encode different antiviral defense systems in bacteria, they are clustered in a so-called defense islands. And there are a lot of different genes that encode different antiviral defense systems in these islands, including like restriction-modification, CRISPR, and others. And these defense systems are very important in bacteria. For example, in bacteria Microcystis aeruginosa, nearly 30% of the genome is occupied by antiviral defense systems. So it just shows you in what kind of hostile environment this bacteria lives, because it has to spend nearly 30% of its genome resources just to encode for different antiviral defense systems. And in my lab, we study molecular mechanisms behind these different antiviral defense systems, and we use a combination of biochemistry, molecular biology, genetics, and structural biology to understand how different antiviral defense systems function in bacteria. And for a long time, we were studying the antiviral defense system in bacteria that is called restriction-modification. And it's a simple antiviral defense system comprised of just two enzymes, a restriction enzyme and methyltransferase. And the restriction enzyme, in fact, is able to recognize a short nucleotide sequence in the invading phage DNA and cut this sequence at specific sites. So restriction enzymes in this respect function as small molecular scissors that cut invading foreign DNA. So the methyltransferase recognizes the same sequence as a restriction enzyme, but catalyzes a different chemical reaction. It is able to bring methyl group on A or C bases, making the cleavage site resistant for restriction enzyme cleavage. So therefore, host DNA becomes protected against restriction enzyme cleavage, while invading foreign DNA is cut by restriction enzyme. So in this respect, the restriction modification system functions as an innate immune system of bacteria because it can act immediately on the DNA that lacks the protective methyl group. So restriction enzymes are very, they recognize short nucleotide sequences like this one shown for ECOL1 in this picture. And they just cut both DNA strands, introducing double-strand break. So in my lab, we aim to understand how different restriction enzymes achieve their specificity and how they are able to discriminate short nucleotide sequence from the vast majority of different DNA sequences present in the bacterial genome. And again, to address this question, we were using a combination of structural, biochemical, and genetic methods. And of course, restriction enzymes, they are very diverse. So there are nearly 4,000 of different restriction enzymes. So the question is, which enzyme you then choose for your studies from 4,000 different restriction enzymes? And this is exactly the same question that you experience when you go to wine shop. So which one you choose for your experiments this evening? So we decided actually to focus on a group of restriction enzymes that recognize related nucleotide sequences. So all these restriction enzymes in this group, they have the concept CCGG tetranucleotide that is flanked by different nucleotide sequences outside of the concept core. And we aim to understand how different restriction enzymes in nature are able to achieve different specificity, how they are able to recognize different nucleotide sequences. And again, we aim to solve the crystal structures of all restriction enzymes that belong to this group, and then try to correlate their structures with function of restriction enzymes. And it was really an interesting exercise. And it took us nearly 20 years to solve these crystal structures of restriction enzymes. And we naively saw that maybe if we'll be able to understand how nature designed the restriction enzymes for different sequences, so we saw that maybe we can, guided by this information, we'll be able to redesign the specificity of restriction enzymes and target them to any sequence in the genome, making them maybe useful even for genome editing applications. And all currently available DNA editing technologies, or most of them, rely on molecular tools that can introduce double-strand break in the DNA. And the DNA the double-strand break in the DNA, it triggers mechanisms of the double-strand break repair that could occur in a cell either by non-homology end joining or homology recombination. In the case of NHEJ, actually small insertions or deletions are formed at the double-strand break site, while in the case of homology recombination, the repair in the presence of the repair matrix is scarless. So in order to introduce a precise double-strand break at specific site, so these molecular tools should recognize long enough target sites in the DNA that could be unique in the genome, and then they have to introduce double-strand break at the target site with high efficiency and avoid cleaving other DNA sites that we call off-target sites. And importantly, these molecular tools also have to be easily reprogrammable that would enable targeting of any site in the genome at low cost. And again, so in the genome editing field, before CRISPR, meganucleases were used for genome editing applications. And there are three groups of meganucleases: homing endonucleases, zinc finger nucleases, and talons. And despite of the differences of the scaffolds of these meganucleases, all of them use a DNA binding domain to read out the specific DNA sequence. And all these meganucleases read their DNA target sequences using hydrogen bonds and van der Waals interactions between protein amino acid residues and donor and acceptor sites on the base edges. And it's important that in the DNA major groove, each base pair has a unique pattern of donor and acceptor sites on the DNA. And proteins are actually able to read these donor and acceptor differences in donor and acceptor sites, making particular hydrogen bond or van der Waals interactions, and in this way they can discriminate different target sites. However, if you think about reprogramming these meganucleases, it's becoming a real challenge, because in order to reprogram meganuclease, you need to replace amino acid residues at the protein DNA interface. And this is a challenging task, because even if you are able to design new variants, these new variants are often prone to off-target cleavage. So in 2007, a paper appeared in Science Journal that claimed that scientists in France and Canada, so they discovered a new antiviral defense system that was called CRISPR. And they claimed that actually this antiviral defense system provided interference against viruses in bacteria. So how the CRISPR-Cas system works, so when phage infects bacteria, so most of the bacteria die. However, a small number of bacteria survive. And it turned out that survivors were able to hijack small piece of invading foreign DNA and then incorporate this piece of DNA into a CRISPR array as a new spacer. And then actually the bacteria can use the newly incorporated spacer as a template to make small RNA molecules that are called CRISPR RNA. And these small RNA molecules, together with Cas proteins that are encoded by Cas genes located in the vicinity of a CRISPR array, they form a so-called effector complex. And this effector complex, guided by CRISPR RNA, triggers invading nucleic acid destruction during next round of infection. So in this respect, the CRISPR-Cas system functions as an adaptive immune system of bacteria, because it's able to memorize the invader by inserting pieces of invader's DNA into bacterial genome. And in my lab, we aim to understand how different CRISPR systems provide interference against invading phages. So we have focused on the CRISPR-Cas systems of Streptococcus thermophilus bacteria. And this bacteria is industrially important bacteria because it's widely used in a food industry, for example, to make cheese and yogurt. And in a specific Streptococcus thermophilus strain, there are 4 different CRISPR systems that belong to the different CRISPR-Cas types, and then actually we aim to understand how different CRISPR-Cas systems in Streptococcus thermophilus functions and provide interference. So of course, when you look at these 4 different CRISPR systems, the first question is which one again you choose for your studies. So it's not 4,000, it's just 4, but anyway, it's still a question. So initially we decided to focus on a CRISPR-3 system because of two reasons. It had the smallest number of genes in a CRISPR system, and then actually one of the genes that encoded Cas9 protein, in fact, it had a signature motif that we've seen before in restriction enzymes. So it was a good hint that maybe the Cas9 could be important in the interference. Yeah, so we didn't know how to work with Streptococcus bacteria, and I'm making jokes that in my lab we didn't know how to make yogurt or make cheese. And so therefore we decided to transfer the CRISPR-Cas system of Streptococcus into E. coli that is actually a workhorse in the lab. And to our surprise, actually, we were able to clone the genes encoding CRISPR-3 system into E. coli, making a heterologous host. And actually, this CRISPR-Cas system in E. coli, in fact, it was functional and provided interference against E. coli phages or plasmids if a matching spacer was engineered into CRISPR array. And then, actually, using genetic tools available in E. coli, we were able to demonstrate that, in fact, the Cas9 protein is the only protein that is required for interference. So I think this experiment was important because it was the first demonstration that CRISPR-Cas systems are transportable. It means that you can take CRISPR-Cas system from one organism and actually transplant it into a different organism. And these CRISPR-Cas systems remain functional. Of course, the next logical step was try to isolate the Cas9 protein and characterize it biochemically in the tube. And Giedrius Gasiūnas, who was a PhD student in my lab at that time, so he was able to isolate Cas9 protein, and it turned out that a Cas9 protein co-purified with 2 RNA molecules that are called CRISPR RNA and tracer RNA. And later, using a set of biochemical experiments, Giedrius was able to show that these RNA molecules are very important in a Cas9 function. So we were able to show that the Cas9 ribonucleoprotein complex guided by RNA molecule, by CRISPR RNA, is able to recognize specific target in the DNA, and then Cas9, using 2 different active sites, is able to cleave DNA, introducing a double-strand break. And this double-strand break then triggers mechanisms of the cellular repair that could be harnessed for gene knockouts or knock-ins. So the simple organization of Cas9 ribonucleoprotein complex, where specificity is provided by RNA molecule and cleavage is provided by Cas9 protein, actually paved the way for developing of new genome editing tools. And indeed, later, Cas9 RNP was widely adapted for genome editing experiments in very different set of organisms. So the Cas9 is a great tool, of course, and it's now rapidly advancing into the clinics. However, there are still challenges that need to be overcome. And actually, one of the challenges is targeting space limitations, because Cas9 protein requires PAM sequence for cleavage, and I'll talk a bit about PAM problem a bit more in a few next slides. And then the next challenge is related to delivery. Adeno-associated viruses are often used for delivery of genome editing tools, especially in human therapeutic application. However, the cargo size that adenoviruses can carry is limited. And Cas9 is quite big protein, so therefore packaging limitations of AAV viruses becomes a problem, and therefore smaller genome editing tools are required. And of course there are other challenges, but in my talk today I will focus on the first two. And of course there are two general solutions to these challenges. First, one can try to engineer Cas9 variants using the ones that are already available. And another approach is just to look at the natural diversity. And in my lab, we focus on the natural diversity approach. So the CRISPR-Cas systems are very diverse, and CRISPR nucleases are also very diverse, and this phylogenetic tree illustrates the diversity of Cas12 and nucleases, and also Cas9 nucleases. So in my lab, we aim to explore this diversity of CRISPR-Cas nucleases, and actually I split the rest of my talk into three different short stories. So in the first part, I will talk about Cas9, exploration of Cas9 orthologs that allowed us to overcome space targeting limitation. In the next part, I will focus on miniature nucleases that allow us to address the delivery problems. And finally, at the third part, I will talk about novel genome editing tools that we discovered recently. And this is a protein that belongs to transposon, and it's first non-CRISPR RNA-guided DNA nuclease. So the specificity of the Cas9 protein is dictated by interactions between CRISPR RNA and matching DNA sequence. So the binding of the CRISPR RNA to the matching DNA sequence actually makes a DNA structure that is called R-loop. However, to initiate binding of the CRISPR RNA to the target sequence, Cas9 protein first has to recognize the PAM sequence that is located in the vicinity of the target sequence. And if there is no PAM sequence, there is no Cas9 cleavage or binding, even if the matching CRISPR RNA is 100% matches the target DNA sequence. So, and the PAM sequence is a characteristic feature of Cas9 protein, and different Cas9 proteins, they recognize different PAM sequences. So, and the PAM sequence requirement, in fact, limits target site selection. For example, the canonical S-pyogenous Cas9 that is widely used for genome editing application, it requires GG PAM sequence that constrains targeting to every 8 base pairs in a human genome. And actually, it makes nearly 75% of mutations associated with human diseases non-acceptable for Cas9 cleavage when we consider very precise targeting. So we reasoned that Cas9 proteins coming from different bacteria actually can help us to solve the PAM targeting space limitation problem that is related to PAM requirement. And Cas9 proteins are very abundant. If you look at the DNA databases, there are thousands of Cas9 sequences. And to identify new Cas9 variants, we actually developed a phylogeny-guided approach, so that allowed us to identify novel Cas9 proteins, and guided by this information, we were able to build a phylogenetic tree that included thousands of different Cas9 variants. And then, actually, we aim to characterize Cas9 variants that belong to the different branches of the phylogenetic tree. And we selected nearly 100 different Cas9 orthologs that are located on different branches of phylogenetic tree and aim to characterize them biochemically. Yeah, but in order to characterize the Cas9 orthologs, you need to determine the time sequence requirement, and actually to get the time specificity for Cas9 orthologs, we developed a simple pipeline that actually that included the expression of Cas9 genes encoded Cas9 proteins in the in vitro translation transcription system. And in the same system, we can also make the guide RNA. And then actually we used the Cas9 RNP that was assembled in the in vitro transcription translation system as a source of Cas9 protein. And we used these Cas9 proteins to cleave the plasmid DNA library that contained a unique sequence targeted by CRISPR-RNA that was flanked by 7 nucleotide sequence in the vicinity of the target site that corresponded to the PAM sequence. And then actually we captured the cleavage products by adapter ligation and determined the PAM sequences that license DNA cleavage in the library by deep sequencing. And this assay, I like this assay because it's a positive selection assay. It allows you to determine only those PAM sequences that licenses DNA cleavage. And using this assay, we were able to establish PAM sequence requirements for more than 50 different Cas9 proteins. So, and we were able to establish PAM sequence, Cas9 variants that recognize T-rich PAM sequences, C-rich PAM sequences, G or C-rich PAM sequences, or combinations of different nucleotides. So in total, these Cas9 orthologs recognized different PAM sequences that differed from 2 to 5 nucleotides in length. So all these Cas9 orthologs that I described and we were able to identify now actually is developed further by Cas9 company that we established in Vilnius. And those of you who are interested in trying these Cas9 orthologs in your own research should contact Cas9. So I just told you how looking at the diversity of Cas9 orthologs allowed us to overcome PAM limitation problems. So in the next couple of slides, I just show you how looking at the diversity allowed us to identify miniature nucleases that allowed us to overcome the packaging limitations. A few years ago, Shmakov and Harrington identified new CRISPR-Cas systems that contained a so-called Cas12F signature gene. And these Cas12F nucleases, they had a conserved active site that was present also in Cas12A nucleases, or CPF1, that is also widely used for genome editing experiments. However, the Cas12F nucleases showed one important difference. So they were nearly 2 to 3 times smaller than conventional Cas12 nuclease. So therefore, we call these nucleases miniature nucleases. So then, actually, we aim to characterize the DNA cleavage requirement of these miniature nucleases. And again, to characterize DNA cleavage requirements, we used the same assay that we developed for characterization of Cas9 orthologs. So in this case, actually, we didn't express genes encoding Cas12F in the in vitro translation system, but actually, we expressed the Cas12F in E. coli and used E. coli cell lysate as a source of the recombinant protein. And the guide RNA was obtained through expression of the CRISPR array. And then we used these Cas12 RNP complexes to cleave plasmid DNA library. And indeed, we showed that these miniature nucleases were able to cleave double-stranded DNA, introducing double-strand break, when a proper PAM sequence was present in the vicinity of the target site. And most of them of nucleases that we characterized, they were specific for T-rich PAM sequences, however, some of them actually showed a preference for also C-rich sequences. So then the next logical question was whether these miniature nucleases are able to do genome editing in human cells. And to address this question, we engineered two different plasmids that contained two different nucleases, Cas12F nucleases, and also they encoded their guide RNA that targeted 3 different target sites in the hex cells. And then we actually expressed this plasmid in hex cells and looked at the indels by deep sequencing in the specific target sites. And these indels are characterization of the double-strand break repair at specific site that was introduced by miniature nucleases. And what we found that indeed one of these miniature nucleases was able to introduce indels at 2 of the sites, but the editing efficiency was pretty low. However, this was the first demonstration that in fact miniature nucleases could be developed into genome editing tools, and very recently, a few papers appeared in different journals that claimed that these miniature Cas12F1 family nucleases can be used for genome editing in different setups, including AAV delivery. And actually, we, with collaborators, demonstrated that these miniature nucleases could be used for editing of plant genomes with high efficiency. So actually, this brings me to a short conclusion from this part of my talk. So I showed you that looking at the diversity of CRISPR-Cas systems, we were able to identify miniature nucleases, and these miniature nucleases, in fact, are able to cut DNA introducing double-strand break if a proper PAM is present, and therefore these miniature nucleases actually are promising genome editing tools in eukaryotic cells, and they could be compatible with AAV delivery. So, and these miniature nucleases are also available at the Cas9 company. So if you look at the phylogenetic tree of Cas12F proteins, and if you look at this Cas12 miniature nuclease family, so you can see that the closest neighbor or closest relative of Cas12F nucleases is a TnpB protein. So what is the TnpB protein? So TnpB protein belongs to the transposons. And transposons are mobile genetic elements that can jump between different sites in the DNA. And the TnpB protein that we focused on, actually, it belongs to the so-called IS20605 family of transposons. And these are actually nearly minimal transposons known to date because they are often comprised only of 2 genes, TnpA and TnpB, and these 2 genes are flanked by left and right transposon elements that are important for transposition. And the transposition of the DNA in this case occurs during DNA replication, and TnpA protein, in fact, is a transposase that is able to excise a single-stranded DNA from one of the DNA strands during replications, and actually this excised DNA makes a single-stranded DNA cycle. And then actually TnpA is able to insert this transposon in other site in the genome, and this site also has a specific sequence. The transposition occurs not randomly, but at specific sites that actually are specified on the top. It's TTGAT site. So the transposon is excised at this site and then inserted again into the same site. So while the role of TnpA was clear, it's a transposase. So what a TnpB protein is doing in this transposon? And this was actually the question that we wanted to address. So we used again this biochemical approach. We aimed to purify the TnpB protein that was part of Deinococcus radiodurans ISDRA2 transposon. And we expressed this TnpB encoding gene in a plasmid together with other plasmid that encoded the whole transposon, except that TnpA transposase was inactivated to avoid gene jumping in the system. And we were able to isolate TnpB protein, and it turned out that TnpB protein, again, co-purified with a single RNA molecule. And then, actually, to understand what is this RNA molecule that co-purifies with TnpB, we actually isolated this RNA molecule and subjected it to the RNA sequencing. And what we found that indeed the TnpB purified this RNA molecule that was about 150 nucleotides in length. And this RNA molecule nearly perfectly matched to the 3' end of the TnpB gene. It overlapped also with right transposon element that is at the end of the TnpB encoding gene, and had a variable 60 nucleotide sequence that was coming from the plasmid. So since this RNA was carrying the coding sequence for a right transposon element, we call this RNA RNA from right element RNA. So next, we decided to look whether TnpB is able to cleave DNA. And we did it using the same approach that I described you for Cas9 orthologs and miniature nucleases. Again, we expressed TnpB protein together with RNA that targeted a specific site in the plasmid DNA library, isolated the TnpB protein, and subjected plasmid DNA library for the TnpB cleavage. And what we found that indeed TnpB was able to cleave DNA introducing double-strand break. And then when we looked at what sequences the TnpB cleaved DNA, and it turned out that cleavage occurred when the matching sequence was close to the TTGAT sequence. And this is exactly the same sequence that is required for transposon to initiate transposition. So therefore, we call this sequence TAM, or transposon-associated motif. So taken together, this experiment showed that TnpB protein, in fact, functions as RNA-guided DNA endonuclease that is able to locate the target site guided by RNA, and this target site should be flanked by a proper TAM sequence. And then TnpB protein is able to cleave DNA, introducing double-strand break. So the next step was just to look whether TnpB could be harnessed as a genome editor. And again, we engineered a plasmid that encoded RNA together with TnpB protein, and this RNA, in fact, targeted 5 different sites in the human genome. And we transfected HEC cells with this plasmid and looked at the cleavage at 5 different sites. And what we found that indeed TnpB was able to cleave DNA at 3 of 5 out of the 5 target sites. At some sites, the cleavage was pretty efficient, so it reached about 20% without any further optimization of guide RNA or the protein. So I think this is kind of first demonstration that TnpB protein could be developed into a new class of genome editors. So if you look and compare TnpB to other genome editors that are available today, so that TnpB has some advantages. For example, it's the smallest genome editor known to date. It's comprised nearly of only 400 amino acid residues. And actually, it cleaves DNA, introducing double-strand break, and therefore it's compatible also with AAV delivery. So this brings me to my summary slide. So during my talk, I actually showed you how Exploring the diversity of different CRISPR-Cas systems allowed us to identify novel Cas9 orthologs that recognize different PAM sequences, and these Cas9 orthologs can be used to overcome targeting space limitation for Cas9 proteins. So again, so I get back to the analogy of scissors. So instead of having a single pair of scissors, now we have multiple scissors that can cut at different sites. So next, I showed you that actually using the same approach, we were able to identify miniature Cas12F nucleases that are already able to cleave DNA at specific sites similar to Cas9, but they have much smaller size. And finally, I introduced you to novel genome editing tools that belong to the transposon elements, and these genome editing tools could be harnessed for different applications. So I think that this toolbox, in principle, could be developed into a plethora of different genome editing tools that could be used for very, very different applications. So finally, I would like to acknowledge people who contributed to this work in my lab at Vilnius University. So at my team at Cas9 and our collaborators at Corteva Agriscience, where we collaborate with Corteva Agrisciences for a pretty long time, developing genome editing tools for plant genome editing, and also our collaborators at New England Biolabs. And thank you very much for your attention.
Thank you, Professor Siksnys, for sharing your interesting work with us. May I request you to remain on stage for the Q&A session. I would now like to invite Professor Tan Meng Hao, Associate Chair of the School of Chemistry, Chemical Engineering, and Biotechnology, to be the moderator for the Q&A session.
Hello? Hello? Okay. All right, great. So thank you for that very fascinating talk. So let's begin with the questions that the audience have. Okay.
Yeah.
Yes.
Can we ask questions in person or do we need to
Yeah, for sure, you're welcome to ask questions, probably in person. No, but.
Yeah, thank you very much. First of all, thank you so much for the insightful and inspirational talk. I have learned so much about CRISPR-Cas9. In fact, the first part of your talk, it brought back memories of me running very big sequencing gel, those days, and then reading the sequence using a ruler. Yeah, so those were the days. Of course, times have changed. And what I want to ask you really is about CRISPR-Cas9 editing of methylated genome. You show that in passing in a very last slide. Whether you think it is actually feasible for CRISPR-Cas9 to represent a strategy to not just alter the genome, but also the epigenome, especially with reference to human diseases, the in vivo feasibility. Thank you.
Yeah. Thank you. It's indeed an important question because you know that many sequences are probably methylated in the genome and carries these different epigenetic sites. And people looked at the cleavage of Cas9 protein at different sequences, and they found that indeed, if the methylation occurs in the PAM sequence that is recognized by the Cas9 protein, so then methylation can interfere with Cas9 cleavage. So if methyl sequences are present somewhere in a DNA sequence, so the effect is much smaller because in this case, recognition occurs through Watson-Crick base pairing between the DNA and methyl groups are not interfering too much to that, yeah. So, but methylation in PAM sequences are important.
Yeah, so the question about, do you see it as a direction, perhaps because CRISPR-Cas9 is now outed to be, I think, one that can revolutionize medicine, right? So in terms of human diseases, practically even possible to use CRISPR-Cas9 to actually modify the IP genome?
Yeah, so of course, the CRISPR-Cas9 protein is a great tool for DNA cleavage, but already in 2012 we showed that if you just mutate active site residues in the Cas9, so the Cas9 stops cleaving. But actually, since binding is guided by the RNA, so it still binds to the target sequence if the PAM sequence is present nearby. So, and this property actually, using the catalytically dead Cas9 protein, people were able to use it as a platform to fuse some enzymes that could just change the methylation status or change the transcription of the genome. And actually these tools could regulate the transcription or epigenetic status of the cell and make an impact on the cell. Yeah, so I think that these tools are also very important because they do not introduce any permanent changes in the genome. So there are no double-strand breaks, there are no off-targets in this case.
Yeah, thanks very much. Yeah, got to raise your hand.
Can I ask you two questions from a non-biotech person? If bacteria are so clever to develop this CRISPR system to defend themselves against viral infection, from an evolutionary point of view, how come humans do not have this system? Do we have something like CRISPR to protect ourselves from viruses or other infective agents?
Yeah, of course. So there are different systems. So there is no CRISPR-like system in human cells. So there is something, yeah. So in eukaryotic cells, there are proteins that are called Argonaute proteins. So these Argonaute proteins work similarly. They are guided by RNA molecule, and these Argonaute proteins can identify invading foreign DNA or RNA. So, but there is no CRISPR system in human cells. But in human cells, there are other defense mechanisms that provide interference against invading viruses. So these kind of sting-related systems, interferon-related systems that allows to recognize invading foreign DNAs, including viral DNAs, and these systems protects human cells against viruses. So that was important during the COVID infection, that people looked very much at the mechanisms, how viral DNA is coming or RNA is coming into cell, and what systems are activated in this case, yeah. Yes?
And to use CRISPR in gene therapy, you repair the mutation that causes certain diseases. And how would that, because the germline mutation actually exists in all the cells of a human person, how would that be able to correct, for example, a person with Huntington's chorea, which has a mutation of not just certain cells, but every cells in his body? How do that work, even if you can repair the gene?
Yeah, so this is an important question, of course. And if you look at the clinical trials that are now going using CRISPR nucleases, so most of them are actually focused on cells and tissues that could be engineered ex vivo. So actually you can take, let's say, bone marrow cells out of the body. So make engineering in these bone marrow cells that are producing, for example, blood cells in your body, engineer on your dish. So then actually takes these cells and put it back into the patient. So in this case, these bone marrow cells, engineered bone marrow cells, will be producing blood cells without mutation. So this is ex vivo approach, or maybe you can take part of the liver, make modifications ex vivo, and then transplant this piece of liver back to the patient. And then actually the liver has a property to regenerate, and then actually these mutations could help to correct the genetic defect or disease that is related to genetic defect. Other approaches that people are using or in clinical trials are just using CRISPR to treat eye diseases or inherited eye diseases that cause blindness, because actually they can inject CRISPR-Cas system directly into eye, and eye is quite isolated organ. So, but then if you would like to engineer every cell in the body, so it's a challenge. So the only way is just to go to germline, but actually this is probably not the way that we would like to go. Yeah. Yeah.
Quick question related to the TnpB.
Yeah.
So where is the promoter that drives this RNA? Is it part of the TnpB gene itself? And the second question is, what's the function of the TnpB in the transposition? Because that was not clear.
Yeah. Okay. So it's a very good question, and I think that we don't know actually whether there is a specific promoter for RNA. So it could be that actually the RNA originates through the processing of mRNA that encodes TnpB gene. So, but actually we don't know how it happens. So it's an interesting problem. And next, actually, what is the role of TnpB in transposition? So due to the time constraints, I didn't show the slide, actually, how we think TnpB functions, but yeah, maybe I can get to my slides. One of the last slides, so maybe I can just navigate. It will be quicker. Going back. No. Yes. Yes. So this one. Yeah. So if you look, actually, as I said, that actually the transposon is excised from a single DNA strand. So then, after replication, you have two different alleles. One allele, after replication, still has transposon, and another allele lacks the transposon. But then, in this case, after cell division, you will have two different cell populations. One of them will be lacking transposon, and another cell will be still carrying the transposon. But actually, what TnpB does, actually, when transposon is excised, this black sequence that corresponds to TGAT site actually becomes positioned close to the sequence behind the blue site. So then, actually, after transposition in the strand where transposon was excised, the cleavage site for TnpB is reinstalled. So then, TnpB cleaves this DNA strand where transposon was excised. And then DNA cleavage triggers homologous recombination, and this homologous recombination reinstalls the transposon copy in the strand that lost the transposon. So this allows the transposon actually to propagate. So, and then the transposon is not lost, yeah, in this case, yeah.
So that happens within the same cell, basically, after replication.
Yeah. Okay.
Okay, all right, so let's finally get to the questions online. Okay, so many of them are kind of revolving around applications, things like what can be the real-world applications for CRISPR-Cas9, how will it change human development, health? But there was a lot of discussion on the function application as a therapeutic. Can you elaborate more on what else can CRISPR do beyond therapeutics?
Yeah, so of course, human therapeutics is an important field, but since actually CRISPR is a universal tool because it introduces double-strand break in the DNA, and the DNA is present in all living organisms. So in principle, you can use CRISPR to engineer all living organisms. So actually, people like to say that now we have tools that allows us to engineer life. But of course, it's important to look at the CRISPR application in agriculture, because actually I think that this very precise editing that CRISPR enables in plants actually makes a lot of interesting applications, because you can engineer new plant varieties using CRISPR tools much more rapid or much more quickly than before, because actually people were doing plant genome engineering from very, very old times. Maybe 10,000 years ago, people started to select varieties, but we know that selection of the different plant variants is very slow process. And CRISPR now enables to speed up the selection, because if you know what genes are related to specific phenotype, you can use CRISPR, insert these genes, and engineer new plant varieties. And this is an important question when we have in mind that climate is changing now very rapidly, and actually now we have to adapt or maybe forced to adapt our crops to changing climate very quickly. And then actually we need these approaches that allows us to do it.
Right, so talking about editing other organisms, so Terry still asks, will CRISPR solve the organ transplant bottleneck, i.e. xenotransplantation?
Yes, so actually people are trying to apply CRISPR, and organ transplantation is an important issue, because many people, you know that many people just die because actually there are not enough donors that can just supply the organs. So, and George Church and other people, they actually started to do genome editing of pigs, and they are engineering pigs just to make the pigs' organs compatible with humans, because then actually, of course, if you just transplant pig heart into human, it can cause rejection because of immune reactions of human cells. So what people are doing using CRISPR, they are trying to just delete the genes that make the antigens on the surface of pigs' organs, and then actually these organs can become compatible with humans. So, and this is already ongoing, yeah, so.
Okay, so we potentially have CRISPR organs in the future. Okay, so let's talk about some of the shortcomings of CRISPR technology. So this question says, are off-target modifications still one of the main risks for effectively using CRISPR technology in humans? Are there any improvements for binding specificity of Cas nucleases in the last few years?
Yeah, so yeah, as I said, that CRISPR proteins and Cas9, they can introduce precise cut at specific location, but sometimes actually they recognize different sequences, and this causes off-target cleavage. And of course, off-target cleavage is undesirable outcome of that, because actually you can correct the mutation at one side, but introduce mutations at other sides, so that's could be also pretty bad. So I think that actually one of the approaches is just to look at other Cas9 orthologs that we and others developed, and these Cas9 orthologs actually maybe they can show less off-target cleavage. And there are other approaches that people are using catalytically dead Cas9 or nicking variants of Cas9 to develop the genome editing tools that does not require double-strand break. So there are technologies that are called base editing or prime editing that allows you to change the heterocyclic base without introducing a double-strand break. And this is maybe a safer technology that could allow these off-target problems.
Okay, so that's on the off-target modifications. So, but that's, there are probably other shortcomings of CRISPR systems. So this question asks, what considerations are made in the development of the bettering of current CRISPR-Cas technologies? And something that comes straight to mind is a lot of the Cas proteins come from human pathogens. So there have been multiple reports that say that humans, many of us actually have pre-existing antibodies targeting, even the SpCas9. So that kind of limits the in vivo applications. So how do you envisage overcoming such a problem?
Yeah, so you are right that actually the Cas9, aspergillus Cas9, actually that is most widely used for genome editing applications, comes from human pathogen. And actually many of us actually have this pathogen in our throats. So it's not surprising that actually then introducing Cas9 from aspergillus, and people demonstrated that in lab experiments can cause immune reactions. So I again think that using Cas9 orthologs that are coming from very different bacteria, not human pathogens, but other bacteria, could help to overcome this immunogenicity of different Cas9, or immunogenicity of Cas9 protein.
Right, so this, there's a question that's kind of related. So does it mean that TnpB, which you just described, can be less immunogenic and cause less off-target cutting?
Yeah, so I didn't have time to tell you, but if you look at the phylogenetic tree of TnpB, actually it's a really stunning variety of TnpBs. And these TnpBs come from very, very different bacteria. Some of them, the well-characterized TnpBs come from H. pylori. So of course the TnpB from H. pylori could be immunogenic, but some of TnpBs comes from like Deinococcus radiodurans. They are not present in our organisms. And therefore this variety of TnpB also can help us to overcome the immunogenicity problem.
Okay, so this, all right, so then this question from, since we're talking about TnpB, this question from Eric Yap. Well, since it's a remarkable discovery that TnpB is RNA-guided, is this likely to be a common mechanism for other transposases as well?
Yeah, so the TnpB is present in a specific site, specific transposons that belong to this family that you showed. So this is IS200, IS605. So other transposons, they have different sets of proteins, and so they function by different mechanisms. So if you look at the TnpBs that are guided by RNA, so most likely you find them in this specific class of transposons that I described. So other transposons, they carry a very different set of proteins.
Okay. All right, so I think many people in the audience are users of CRISPR technologies. And one of the things that's maybe graduate students and postdocs are bewildered by is the sheer number of scissors that you can choose from. So there's this question from Mandy. How is the performance of TnpB compared to Cas9 or the Cas12? Has your lab done a side-by-side comparisons? And then to add on to that comparison of the different nucleases. So just now relating to Carl-Leon's question, you can fuse, for example, DNMT3A to metallate and so on. But a lot of the work was done on Cas9, SpCas9, which is actually very big, right? And then so would the same strategy of fusing all these different domains, the aminases, gene activation domains and so on, work for a small protein like TnpB, right? So SpCas9, you can imagine it to be like a lorry, right? A big lorry can carry big cargo. And now you have developed a Porsche, which is great, right? Porsche is small and fast, but can it also be fused to big domains like Viper and the Appleback, the aminase, and still work? Yeah.
Yeah. So following your car analogy, so probably we're trying to develop not a Porsche but Mini Cooper, where you can attach the big part of it. Yeah. So, but yes, of course, this strategy should be valid, and these experiments are currently ongoing in my lab. So what we're doing right now, we are trying to look at the diversity of TnpBs, because the TnpBs that I showed you, actually, it recognizes a pretty long time sequence, so that could be a limitation if you think about addressing some target sites. But then, actually, it could be that different TnpBs may show different time sequence requirements similar to Cas9 proteins. So then, actually, looking against the diversity of TnpB, you maybe can find TnpBs that shows requirement for very short targeting time sequences. Yeah, and then actually you can use the catalytically dead TnpB, and we made the catalytically dead TnpB just mutating amino acid residues at the active site as a platform for binding to that target. And then actually you can attach different effector heads like deaminases or methylation enzymes to it. And that's kind of could be developed into epigenetic tools.
I see. So this is work that's ongoing in your lab
Yep.
right now. Okay, that will be interesting to see what the results will be. So I was pretty sure that this question will come out, and indeed it did come out. Something about, how far away are we from a future where babies can be genetically modified to have the best traits?
Yes, the chairs already discussed this question and this problem. So it's kind of, it's a problem. But when I hear this question, so always people are saying, oh, best traits, but what is best? So if you look back at the history, so the perception of what is pretty or nice, so it changed a lot. So then actually, if we stick to best traits, so that could be a problem. But of course, the important question is whether we can use this approach to treat human diseases. So, of course, now in the CRISPR and genome editing field, so there is a red line that actually people are not doing any embryo experiments. But this question probably inevitably will come up in the future. And I think that it's very important also to engage in the discussion with scientists and non-scientists and with society to know the perception of society, how society finds it. Is it possible or not from their view? And actually, I think that important with society is very important. And it's differently what Yang Hui did, actually. He did this experiment, not informing other people what he's doing, and it came out of the blue. So this is not the way how scientists should do it. Yeah.
Okay, I think we are almost, actually we are out of time. So I'd like to close with one last question, because I know there are a lot of students in the audience and tuning in online. So what do you think are the qualities of a great scientist? And let's say if you are an 18-year-old teenager right now, what advice would you give yourself to pursue a career in science?
Yes, of course, always risky to give advices. So. But I think that the passion is important in science, because you should try to find the job that somehow motivates you. So in the work of scientists, I often say for students that science is not a job. It's a kind of what motivates you, and that allows you to stay long hours after working hours, because you are just driven by something that is interesting, and that motivates you a lot. So the passion in science I think is very important. So you should like what you are doing. And if you don't like that, maybe you should think about other things and other paths in your career.
Okay,
sorry,
so I understand that, and people who want to ask questions.
Sorry. Can I ask a question? So I'm a college student, right? So then, may I know how do you become a professor, and do you have any advice for students who aspire to be scientists?
Could you? Yeah, okay.
May I know, like, how do you become a professor, actually? Like, can you just give a brief journey? And do you have any advice for students who aspire to be scientists in the future?
Yeah, so the question was how I became a professor?
Oh, like, how do you become a professor? And the journey you took towards it?
Oh, okay, so I would say that actually I was never driven by the desire to become a professor. So just I was driven by experiments that we were doing in the lab and what interested me. So then the professor comes like a side reward, yeah. So but it's not a major reward, yeah. So.
Yeah, but what are the steps you take to work towards your goal, your job? Like how do you, what do you do to get to where you are now, and your job of discovering biology?
Yeah, so in fact, this is what motivates you. So this kind of science is a long journey. So and when you're doing science, you should be very patient, because some people are saying that doing science is just moving from one mistake to another, but at some point you find something that actually pays off, and that's kind of a real joy of discovery comes then. But you should be very patient.
Actually, what I meant is, what do you study? How do you get to your career?
Oh,
yeah.
Okay.
Yeah, the career progression.
Yeah. So I started I graduated as a chemist. So I never thought I'd be just doing genome editing or talking about genome editing. But I started as a chemist. So and then actually I also changed fields, moving to different areas of research. So I graduated as an organic chemist. So then I moved to study of enzymes that cut proteins. So then restriction enzymes, and then CRISPR, and now moving to other antiviral defense systems. So actually changing fields is important thing. So because actually it makes your life interesting, because no one wants to do the same job like a robot for the rest of your life. So that's kind of, yeah. So I think that you should take your path and be adventurous enough just to change fields, yeah.
Okay, I think that we should stop here. So let's put our hands together to thank Virginijus again.
May I request Professor Siksnys and Professor Tan to remain on stage for the photo taking, please.
Yeah. See it? Okay.
I would now like to invite the following guest to join the group photo taking. Professor Joseph Sung, Professor Peter Preiser, Professor Lim Ka Leong, Professor Sam Zhi Qian, Professor Chiba, and Professor Chen Peng. Thank you, professors. May I request Professor Sung and Professor Siksnys to remain on stage for the memento presentation. And may I invite the other guests to take their seats. Thank you, Professor Sang and Professor Siksnys. May I invite you to take your seats. This brings us to the end of the public lecture. I'd like to thank everyone for your kind presence and those of you with us online. May I request all to be seated while our speaker and senior management take their leave first. Lastly, we request all attendees to fill in the feedback form by scanning the QR code on the screen. You may wish to indicate in the form if you need the endorsement for your attendance. Thank you once again for taking the time to join us today. Have a lovely evening ahead.